Welcome to Immunopolis, a bustling, well-guarded city where immune cells act as vigilant patrolling citizens, sharing intelligence and launching coordinated defenses. Through our journey together in this city, we will explore how the immune system operates as a sophisticated defense network, protecting the body against a wide range of pathogens and the development of cancer. The immune system is divided into two complementary arms working in harmony. The first arm, innate immunity, acts as the vigilant frontline guard, responding instantly and broadly to any intruder. The second arm, adaptive immunity, follows as the intelligent strategist, tailoring a precise response and remembering the enemy for the future. We will begin by illustrating the key barriers of innate immunity. Physical and chemical innate barriers form the body's first line of defense against invading pathogens by preventing their entry and survival. Physical barriers include intact skin and mucous membranes. Chemical barriers work alongside these structures, such as the acidic pH of the stomach and antimicrobial peptides and enzymes. Together, these barriers create an environment that is hostile to pathogens, limiting colonization and infection before deeper innate immune responses are activated. Now let's talk about our invisible allies, the microbiota. These are communities of beneficial microorganisms that normally inhabit the skin and mucosal surfaces, where they protect us by several mechanisms and support immune homeostasis. When first-line barriers are compromised, the innate immune cells spring into action immediately. These primarily include neutrophils, macrophages, natural killer cells, and dendritic cells. Together, these innate immune cells provide rapid, non-specific protection while also shaping subsequent adaptive immune responses. They detect pathogens and tissue injury through germline-encoded pattern recognition receptors that recognize conserved microbial structures and danger signals known as pathogen and damage-associated molecular patterns. Neutrophils act as fast-acting killer scouts, rapidly recruited to sites of infection to destroy invading microbes. Macrophages function as large professional engulfers, eliminating pathogens while releasing inflammatory mediators. Natural killer cells target and eliminate virus-infected or stressed cells. Dendritic cells serve as immune sentinels, capturing antigens in peripheral tissues and migrating to lymph nodes to activate naive T cells, thereby providing a critical link between innate and adaptive immunity. In response to the release of pro-inflammatory cytokines, a robust inflammatory response is rapidly initiated. This innate immune reaction functions to eliminate the source of damage while promoting tissue repair. It is characterized by the coordinated release of inflammatory mediators that induce vasodilation and increased vascular permeability, enabling plasma proteins and leukocytes to exit the circulation and accumulate at the affected site to contain and resolve the insult. When innate immune barriers fail to fully contain the insult, it is time to call in the professional backup. Dendritic cells capture fragments of the invading pathogen and migrate to the regional lymph nodes, where they present these fragments, known as antigens, to naive T cells. This critical step marks the initiation of the adaptive immune response, transforming an early, non-specific defense into a targeted and highly specific immune attack. Adaptive immunity comprises two complementary arms, the cell-mediated and the humoral responses. Activated T cells orchestrate cell-mediated immunity by secreting cytokines that coordinate and amplify immune responses, and by directly eliminating infected or malignant cells. In contrast, activated B cells drive humoral immunity by proliferating and differentiating into plasma cells that produce antigen-specific antibodies, which neutralize pathogens, promote opsonization, and activate complement. After the danger has been cleared, regulatory mechanisms are engaged to restore the immune system to its resting, steady state. This process, known as immune contraction and homeostasis, involves the controlled elimination of excess effector cells and suppression of inflammatory signals. It is essential for preventing prolonged or excessive immune responses that could otherwise lead to tissue damage and loss of immune balance. A small pool of memory B and T cells is preserved, ensuring long-term protection while maintaining immune balance. Immunological memory is a defining feature of adaptive immunity that enables the immune system to respond more rapidly and effectively upon re-exposure to the same antigen. The innate and adaptive immune systems work in close collaboration to provide an effective and well-coordinated host defense. Innate immunity delivers rapid, non-specific protection and supplies the essential stimulatory signals required to activate adaptive immune responses. In turn, adaptive immunity strengthens innate defenses, creating a powerful positive feedback loop. This bidirectional interaction ensures efficient pathogen elimination, precise immune regulation, and long-term protection through immunological memory.